How's it going? Dr. Tyler Pansner, and this is the Genetics 101 course. This is a ground level course to take you from knowing basically nothing about genetics and DNA for you to better understand why this is so immensely important and how better understanding your genetics can give you a whole new layer of understanding yourself to dictate and help you understand what you do and don't need to feel your best. If you haven't already, I would recommend watching the cell signaling model of disease course prior to this. You'll still be able to grasp this, but that course is more so aimed at how cells work in general. Now we're zooming in a little bit more about how genetics work. So why should you listening right now even care about genetics? Let's dig in. So to start off, we are all made up of around 30 trillion cells. That's a T. That's a lot of zeros. For every star in our galaxy, we have 400 cells in our body. So you listening right now has 400 times more cells in your body than there are stars in our entire galaxy. Each cell has around 25,000 genes. Now we cover more on the genes in the Science of DNA module, but briefly here's our DNA. Genes are specific segments of DNA. Transcription factors, we cover this in the cell signaling model of disease course. These make RNA, which are basically instructions on how to make proteins. Every protein in our body is made by a gene. The gene can be mutated or misspelled, and that protein may not function how it should. But proteins are the workhorses of our cells. Whether it's to make testosterone or make dopamine or fight off a virus or build new bones, these are all done by proteins. They do all the countless tasks in our cells. So DNA, these genes, are the instructions on how to make the workhorses of our cells. And each gene, each protein is like a cog in the machine that makes you, you. So if you have a messed up cog over here, they all are interconnected in some way, and it could affect this cog all the way over here. So who can benefit from understanding their genetics? A couple of facts here. Every single human is made up of cells. Every single human has millions of genetic mutations. So the question isn't, will I get information from genetic testing? Everyone has millions of mutations. That's not up for debate, including many thousands upon thousands of mutations that can be supported naturally. Another fact, every single disease has a genetic component. This is taken straight from the National Institutes of Health, the NIH website. All diseases have a genetic component. However, different diseases can have a different genetic component, meaning not every disease is 100% genetic. Some are more minorly genetic, some are more majorly. An example would be Huntington's disease. It's a neurodegenerative disease. If you have the hunting gene mutation, you are guaranteed to get Huntington's disease. Attention deficiency hyperactive disorder, ADHD, that is around 80% heritability, meaning a lot of ADHD cases are genetic. So this is why better understanding your genetics can give you insights how to feel better. And every single disease starts years or decades before your diagnosis. So I touch on this briefly in the cell signaling model of disease course, but check engine lights. These diseases usually start by more minor symptoms. So these are some examples of some check engine lights. More minor symptoms we accept as normal. Headaches, the aches and pains, the bloating, the fatigue. These are the check engine lights. Something's wrong. Your cells are crying out for help. That's why the light went on in the car, right? Now, what should you do? You should probably go to a mechanic, look under the hood of the car, and get it sorted out so the light goes off. Every disease has a genetic component. Every symptom or ailment has a genetic component as well. If your minor symptoms are the beginnings of your disease, because we need to remember, if the check engine light doesn't get checked, what happens? The car breaks down, the car explodes. So if you don't address your check engine lights now, these things are not normal, people. These are the beginnings of disease. You don't just wake up with a disease. It happens over years and years of the check engine lights from the cell pathways not being in balance. Therefore, every single human being can improve their quality of life through proper, the key word is proper, genetic education. We'll talk about why a lot of this education is not proper in the space nowadays. So when it comes to genetics are just one part of the optimal health equation. They're not everything, but they're certainly not nothing. While genetics can play a larger or smaller role in each person's health, every single health issue has a genetic component. Let's just say the blue here is the genetics. 3 different people. This person, it's 19% of their issues. This person is 35. This person is 14. These are arbitrary numbers here, but my point is if we have diet and exercise and stress levels and healthy relationships, and then there's also genetics. We all have the slice of the pie that is genetics here. And what I like to do with the clients I work with one-on-one is remove the slice of the pie that is genetics. How much better can you feel if we support the genes that need to be the most supported for you? Now, as I mentioned, every human has millions of mutations. So proper genetic testing and interpretation will always provide you with results. So it's not if you're going to get useful information, it's how much will this information help you, how rapidly? Because we're looking at things that are truly genetically disrupted in your cells. But again, it needs to be done properly. You also want to match the exact disrupted step with the treatment. More on this later, but my point is, if you have a harder time activating, turning nutrient A into nutrient B, you can't do that as well. We should give you nutrient B, give you the exact thing that the body has a harder time producing. So, why are so many people let down by genetic testing? It can get a lot of hate. People can doubt it. People say it's not advanced enough to give proper insights right now, and I think that's completely wrong. So 4 main things here. We'll go a little bit more deeper into each. Not looking deep enough, overwhelmed with fluff mutations and reports, a lack of supplement knowledge, and a lack of proper patient education and implementation for their genetic plan. So first off, let's go over not looking deep enough. If you get a genetic report done and it checks 40 mutations, that is not remotely enough. A single gene can be mutated in thousands of different areas. So if you have 40 mutations across many different genes, you're getting a tiny little piece of the overall pie here. A few dozen mutations does not cut it. It's often not if a gene is mutated, but how mutated is it? What I mean by this is, here's the DNA that makes a protein, right? This DNA has no mutations. The protein has no mutations. The protein acts 100% how it should. This person has a mutation, so the protein has a mutation. It only works 80% as well. This person has 2. It only works 60%. This has 3. So my point is, from having 0 to thousands of mutations, where do you fall on that spectrum? If a genetic test tells you, they check you for 1 mutation in a gene, and they tell you, you have 1 mutation, you may be freaking out. Oh my God, this is horrible. But what if you look deeper? Let's just say a panel will test 1 mutation in a gene. Let's just say I look at 15 in that gene. If I'm used to seeing 3 or so mutations in that gene, and you only have 1, it's actually not a big deal, because you actually have less mutations than the normal person does. But what if I'm used to 3 and you have 15? Oh my God, that is a massive, massive weak point for you, but it's something we can support. So my point being here is you need to get data on a lot of different mutations, especially mutations within the same gene. A lot of these companies, I don't know if they're trying to scare people, but it's just, oh, you have this mutation that's linked to this issue. You freak out. But if 1 mutation may not be the end of the world, you need to go deeper. Now, this may seem kind of counterintuitive here, but on one end, I'm saying we don't go deep enough. I'm talking about these fluff mutations here. So this might be you when you got a genetic report done. It's 50 pages. How in God's name do you know what's the most important out of that report? More reports, more data for the person viewing it, for you, the consumer, does not equal better. There are only so many genes that you can support. There's only so many supplements you could take. I believe there's a massive lack of guidance as to which of these genes are the highest priority for you. If someone tells you you have mutations in a report over 50 pages in 700 genes, how are you ever going to support all of them? The catch is you can't. How do you know which few to support will help you the most? There is no real guidance given to the highest priority. It's, oh, here's your big list of information. Go and figure it out. People leave more confused and scared. So it's an interesting balance between you have the analysis side. You want to go deep with your analysis on, the doctor side of things, the software side of things. But then when you report it and show it to someone, especially if they're not a doctor, how do you make sure not to overwhelm them? I believe it comes down to, this is how I coined it, too much data, not enough insight to connect the dots for people. A lot of these companies, they want to cram more and more mutations in, more reports. We give 500 reports. We give 1,000 reports. Who's going to read all that? You don't want to read a dictionary of genes. You just want to feel better. Now, the next issue with the space I see is a lack of supplement knowledge. And I'm in a unique perspective to speak on this because I am a pharmacologist by training. I am a supplement expert. A lot of people know genetics, but how many people know the pharmacology you need to properly treat the genetic mutation? So what I'm saying is you have XYZ mutations. You may read a report that says, take these supplements. How do you know those supplements aren't clashing with any other genes? It's like you're trying to build a house when you know the exact plan, the exact materials, but you have no idea how to use your tools. You don't know how to use the supplements properly. I see this all the time in my practice. People, whether they come from a coach or they got a genetic testing done, they're taking recommendations that are data-driven based on the genes. And these are in quotes because it's a surface level understanding and they're making themselves feel worse. The thing is here, most issues arise from functions of supplements or mechanisms of supplements that aren't advertised. Most people think supplements do one thing. It's a violent example, but let's just say it's a knife. In reality, most supplements are Swiss Army knives. So you may think you're just getting the knife. You may think you're just helping your inflammation. But what if that supplement also pulls iron out of your body? That can mess you up. What if it has a lot of sulfur? That may also mess you up. So you think you're taking things that are helping you. It's helping one thing, but it's making two other things worse. This is a massive issue in the genetic space. All the genetic information, all the supplement recommendation, but there's no guidance. There's not proper education about how the supplements work. It's like a doctor prescribing medications when they weren't trained on how the medications function. It's wild when you think about it, how kind of hands-off Wild West the supplement space is. And it shows with how many people are injured or made sicker, wasting time, money, and time feeling healthy on the wrong supplements. Now, the last one here is the lack of proper education or implementation. So this isn't so much just about supplements, but a genetic-based protocol should be so much more than here are your pills, take them, or here are the foods, eat more of those foods. It should be more than this is what this gene means. Reports and genetic consults are given by people that may know genetics, but they don't understand cell signaling. So this is an important graph here. We have data, information, knowledge, insight, wisdom, impact. Data is just a bunch of information. Sorry, data is just scattered, information. I think I described these two here. So information is when you can kind of label them. Knowledge is when you learn to connect the dots. Insight is when you can figure out, once you connect the dots, which are the most important. And wisdom is when you can tie this all together and make an impact on others. So a lot of these genetic companies that give consults, they only know the genetics. They're not connecting it to the overall cell signaling. And from the cell signaling model of disease course, you probably have a better idea about why this is so important. Education about the big picture of these cellular pathways. You should be educated. What food, it's not just what supplement could help the gene. What foods, lifestyle habits, and supplements can help that gene or that pathway as well as hurt that pathway. I like to say you need to learn the language of your body. You need to take this genetic information and learn how to navigate life with this new blueprint. Not just saying, I can't have this supplement because someone said so. Why can't you? And if you take that supplement, what foods may it clash with? So you need to properly educate and help people implement it better. This is why a lot of people that come to work with me, they have 5 different genetic tests. They're all confused because no one's giving proper education or implementation. So this is what I like to call the simplified theory of disease. All disease is caused by only 2 things. Number 1, the body is exposed to a toxin that interrupts normal cellular function. So a toxin, something that shouldn't be there, shows up and messes up how your cells behave, messes up the cell signaling pathways. Number 2, the body is missing an essential nutrient that disrupts normal cell function. So you're missing what should be there. So either something is there that shouldn't be there, or something that should be there isn't there. And now genetics affect both of these. And every single cellular job inside of your cells is carried out by proteins, which can be mutated to work better or worse. So, in summary here, every single human has millions of mutations, including many that can be supported naturally. Check engine lights are the minor symptoms that many accept as normal, and these are the beginnings of disease. Genetics play a role in every single disease, as well as the check engine lights that eventually turn into the disease. And finally, the current state of genetic testing really falls short in multiple aspects, which casts doubt on the science and the space overall. And it's really just pitiful to see overall because this really has life-changing information. So that's enough about that. I hope you understand now why you should be caring about understanding your genetics. Dr. Tyler Pansner here, and this is the Genetics 101 course. Let's dive a little bit deeper into the science of DNA. So what is DNA? DNA, also called deoxyribonucleic acid, is an organic chemical that contains genetic information that serves as instructions for our cells to make proteins. So not going to go too crazy deep into this, but here we have DNA. It's in a double helix type structure you may have heard of before. And the key feature here are the letters, also called nucleotides. So this is the DNA here, and we have cytosine, guanine, adenine, and thymine. C, G, A, and T. These are the 4 letters, the only 4 letters that make up our genetic code. The differences in these nucleotides, in these letters, are what make us each individually unique. Now, DNA isn't stored in our cells just as DNA. We have a lot of DNA. We'll talk about exactly how much in a little bit, but DNA are coiled up in our cells. So how is DNA stored in our cells? Well, DNA is stored in what's called chromosomes. So DNA, here's the DNA. Imagine you're winding DNA around something called a histone protein. And over time, these histone proteins supercoil even more. So a chromosome inside of our cells, this is actually pictures of someone's chromosomes. We have 46 chromosomes in total. These are hyper-coiled DNA. So humans have 2 sets of chromosomes. You get 1 set from each parent. So you have chromosomes 1 through 22. You get half of these from mom. Chromosome 1 comes from mom, 1 comes from dad. 1 comes from mom, 1 comes from dad. Then you have X and Y chromosomes. These are the sex-specific chromosomes. Females get 2 X chromosomes. Males get 1 X and 1 Y chromosome. Now, I said supercoiled, right? How coiled does supercoiled really mean? So DNA, different levels of organization. I'm not going to go into each of these in specifically, but my point is DNA gets wrapped up. It gets wrapped up again. It gets wrapped up even more. It gets wrapped up again. And then it gets wrapped up again. So If you lined up all of your chromosomes, so if you took all these chromosomes and laid them out end by end in from one of your cells, it would be approximately 200 micrometers in length. So that is about the length of this paramecium bacteria. Now, this is obviously not to scale, but this bacteria that we need a microscope to see, if you lined up all your chromosomes end by end, that's how long they would be. Now, if you took those chromosomes and you unwound them, uncoiled them into the DNA strand, so instead of it being condensed, you unwind it. If you lined up all of your DNA, how long do you think it would be? Again, 200 micrometers, the size of a bacteria, is what it's like when it's coiled up. How long do you think it would be? Would you be able to see it with the naked eye? Maybe it's a foot long. Maybe it's 10 feet long. So a fully unwound human genome is around 1.86 miles in length. So each and every one of your cells has almost 2 miles of DNA inside of it. And when you multiply that by the 30 trillion cells we have, that is like, what, almost 60 trillion miles of DNA in each one of our bodies. Pretty wild when you think about it. So that basically means that your DNA is 15 million times shorter when it's condensed. You go from almost 2 miles to 200 micrometers when you coil it up. I thought it was some really cool math to illustrate how really condensed our DNA is and how much DNA we really have. This is really important here. This is what's called the central dogma. This is the explanation of the flow of genetic material within a biological system. So what I mean by this is we have our DNA, and DNA, when we make more cells, we need to duplicate the DNA. More on that in a bit. But DNA, we may have heard from the other courses, the cell signaling model of disease course or the other modules here, proteins here are the workhorses of the cells. DNA gets transcribed into RNA. This is like another set of instructions. Imagine you have to copy it from English to Spanish, and then you got to convert it again into something else. So it's a different set of instructions that then gets turned into proteins. DNA is the instructions to make RNA. Proteins are synthesized from this RNA and are the workhorses of our cells. Genes make proteins. When the gene is messed up, when the DNA is messed up, the protein may be messed up. Proteins are what's required to complete the countless tasks in our body, like grow, adapt, survive, make us feel good, make us feel like crap sometimes. So these are the workhorses. These are what really matter. Now, what is a gene? A gene is a segment of DNA that describes how to make a certain protein. So this segment or region of DNA is also called a loci. Loci can contain single or several genes. It's the basic unit of inheritance. So my point being, the eye color gene. This is a gene that's located in a certain area on the chromosome, a certain segment of DNA. You have DNA, and this segment right here is what dictates what color the eyes are. This is a specific gene that dictates what color hair you may have. Genes can vary greatly in size. We have around 25,000 genes in each one of our DNA, in our genome. Genes can vary greatly in size, from either a few hundred nucleotides or letters long to more than 2 million nucleotides or letters long. Now, how is DNA oriented? I want to touch on this briefly. DNA strands are parallel, meaning you have one strand here and one strand here. They run alongside each other. But they're also something called anti-parallel, meaning one strand is flipped. So here is the two DNA, and here's where you bridge them. This is the two sides of the helix that twist. We're not going to go deep into this here, but basically you have different ends of the DNA, the top and the bottom, the front and the back. So if you look, the DNA, instead of it both being this way, one goes that way and one goes this way. And I say this because when you sequence DNA, you need to rip it open. You rip this open right here, and you read either this side of the DNA or that side, that strand of the DNA. So I've had this question a lot with people I work with, and this is actually an email from a private client of mine that she said, here's my genetic information from 2 different companies I had checked, and all the results are different. What is going on here? So your genes, your genetic mutations do not change throughout your entire life. So this person was asking, this RSID, these are individual mutation names. This is the genotype. So one company told her she has a CC. The other one told her she has a GG. None of these match up. She's like, this is a bunch of crap. One of the companies is lying to me. I'd explained to her, the strands are anti-parallel. So if these 2 were 2 strands of DNA, we flip them open. See how C always matches with G and A always matches with T. So when we split this open, we have a G on one end, right? And a C on the other. We have a T on one end and A on the other. So when you're reading the genes, depending on which strand you read, and it's a random chance during the chemical reaction in the process, you see G and C are interchangeable. A and T are interchangeable. So this is the exact same information. Anywhere where there's a T, you could also write an A. So these 2 companies just reported it differently, but it is the same exact information. Now, if she had a TT and a GG, that's a big issue. If she had a AA and this was a GG, that's a big issue. Just wanted to put that out there, this anti-parallel orientation, and this dictates how it can appear based on different types of genetic company reports. So yes, A equals T and C equals G based on the company. Now, what is a mutation? A mutation is an alteration in the nucleic acid sequence of an organism. So the letters I mentioned, right? And there's 2 main types of mutations. The first is a gene mutation. So a mutation in a single gene or segment of that DNA. Point mutations are changes in one or a few nucleotides or letters within a single gene. Single nucleotide polymorphisms or SNPs. This is the main mutation I like to focus on in my practice. This is when a single letter is misspelled. So instead of an A, you may have a T. Chromosome mutations are larger scale changes in the entire chromosome that can affect many different genes. Not going to dive deep into this, but some quick examples. You can copy and paste a whole extra part of a chromosome. It's not just one gene, it's many genes involved. You could take the genome and you could flip it. You could invert it. You could delete a whole section. You could take instead of copy and paste in the same chromosome, you could take it from this chromosome and put it in that chromosome. My point being, this affects many, many genes and can usually have a lot more profound effects. An example of this is Down syndrome. So Down syndrome is an example of a chromosomal mutation. So here are the chromosomes, right? And what we see here that's interesting, there's 3 chromosome 21s. So the 3rd chromosome 21, instead of having 2 of these genes, now all of your cells are expressing even more genes from that chromosome 21. That's what can lead to the issues associated with Down syndrome, whether it's the facial structure or intellectual capacity or other health-related issues. I want to make this clear. Not every gene or mutation can be supported or bypassed or are hackable with natural things. Some genes, you're just stuck with them. And my point being is Down syndrome is not a genetic disorder that I could say, hey, I'll give you a supplement to support that Down syndrome gene, because there isn't a Down syndrome gene. Down syndrome is copy pasting thousands of other genes. So it's not just a drop a supplement in and they can go and fix everything. This is why we make medications. We can make drugs that are shaped a really special way to try and fit a certain area in the cells to fix disease, but I'm not going to go deep into that. So what is a SNP? A SNP is a single nucleotide polymorphism. As I mentioned briefly earlier, this is a single change or misspelling in a letter within the DNA code. I want to make this clear. A SNP Just because you have a misspelling in one letter in your DNA does not guarantee the protein that's made from that gene will function any differently. Meaning, a mutation in the DNA does not guarantee it'll actually have an effect on your health. I like to call that a silent mutation or a silent SNP. All of the SNPs that I focus on, and most of these genetic companies focus on, are SNPs that do have an effect. I mean, why would you waste time analyzing a gene mutation that doesn't mean anything, right? So on these reports, you'll see genes and mutations that are linked to issues, but just so you're aware listening now, not every misspelling in a gene means the protein is misspelled. That's a whole other can of worms, but just trust me on that. SNPs are the primary mutation I like to focus on in my practice because we all have them and they're very, very well studied. Now, what is an allele? An allele is an alternate version of a gene that makes people look or behave differently. So, here's 3 people here, and here's their DNA. This is the same exact area of the DNA. They all have a different SNP here. These can be 3 different alleles for a given gene. So, for example, a gene, you can have a gene that dictates eye color. But what is an allele? I have the blue eye allele. Someone could have a brown eye allele, a green eye allele, a hazel eye allele. So the gene, the SNPs, can play a role in the allele. But the allele is explaining what it actually does to make them look or behave differently. Now that we know what mutations are, how do mutations actually occur in our bodies? So the first one here, you may be more familiar with this one, are inherited mutations. These are mutations that are passed on from parents to their children generationally. So when we say, I have depression in my family, my grandmother had it, my father had it, I have it, my children have it, that's because this gene is being passed down generationally. You had the mutations in your DNA, and your sperm or egg have that mutation and then create a new organism, a new child that also has that mutation. Now, the other type is something called de novo mutations. De novo means from the Latin phrase over again or anew. These mutations can appear spontaneously on their own. So this means my parents may not have this mutation, but I may randomly get a new mutation. So this is where mutations initially come from, because these inherited mutations, sure, you keep passing them down over and over and over and over again, but how did they initially happen? These are how new mutations enter a population. This is due to issues with DNA replication. We'll touch on that briefly in a few minutes, as well as something called mutagens. These are things that mutate your DNA. So what are some types of de novo mutagens? So things that can mutate my DNA in a way that my parents didn't have done. The first is radiation. So ionizing radiation, so like nuclear fallout, gamma rays, UV light, so like skin cancer. So here's an example here. Skin cancer forms because UV light hits your skin cells, and it can make the 2 T's form something called a thymine dimer. Long story short, this really messes up the DNA. And this is what can initially lead to things like cancer or x-rays. This is why they put a big lead vest on you when you go to the dentist. So different levels of radiation have different risk factors. Then we have chemical mutagens. Asbestos, something used in buildings decades ago. Now we realize it causes cancer. Formaldehyde, vinyl chlorides, coal tar. This is why coal miners can have higher rates of cancer. Tobacco. This is why tobacco can be linked to lung cancer. Alcohol. So here's alcohol here. Alcohol gets metabolized into something called acetaldehyde. This causes DNA damage and can lead to things like not just cancer, but also fetal alcohol syndrome. So this is why if a woman is pregnant with a child and consuming alcohol, the child can die because the child's DNA is being so messed up and mutated from the alcohol. The last one here is infectious. So viruses can take their own DNA and put it into your DNA, which alters your genetic code, which can be passed on. And the overall thing I want to mention here, mutations are going to occur. Our cells get these mutations fairly frequently, but we have repair mechanisms to fix the broken DNA. So if your repair is better than your mutations, you're good to go. You're going to fix what gets messed up. But if your mutations overwhelm your repair system, that is bad. That's when cancer can begin to form. So let's talk about DNA replication. So when you're a cell and you want to split and grow into two separate cells, you got to copy your DNA, right? You can't just have a cell that has all your DNA and a cell that has none of it. So this is the replication process. You double your DNA, you divvy it up, and then you have two daughter cells or two versions of you. So one cell turned into two. This is how we grow as organisms. Duplicating this DNA, when you're copy pasting it, you have enzymes that are doubling your DNA. They're taking all the A, T, C's, and G's, all these letters around the cell, and they're organizing them. Oh, there's an A to A to A, T to T. Sometimes it messes up the ordering. Now, when this happens, your cells, other machinery comes into play. Other workers, other proteins come in, and they can white out. Imagine you made a mistake when you're writing something. You have some white out, and you could write over it. Your cells can fix these errors. So your cells are making errors pretty frequently when we're doubling our DNA, when we're duplicating our DNA, but we have a repair system that then goes in and fixes any errors. And if there's a cell with an error and we can't get it to fix it, your body will tell that cell to self-destruct. Get rid of that cell. We don't even want to risk it acting all weird with those mutations. Mutations, because we tell that cell to self-destruct. We don't need that cell. We have plenty of other healthy cells. Get rid of it. They don't even want the headache. Mutations your cells get during your life only matter if they're not repaired. If they're not repaired and if the cell doesn't self-destruct. What I'm saying is when a cell has a DNA error and it doesn't self-destruct, that is now an early stage cancer cell. And then that cell grows and mutates more and forms a tumor, and we know how cancer goes. So again, we all are getting these mutations as we live. But as long as we can repair it better than we mess it up, we're good to go. But once the mutations overwhelm your repair, you can't repair good enough. This is when cancer can begin to form. So important concept here. Do mutations guarantee issues? If you have a mutation, are you guaranteed to have problems? The answer is no. Definitely not. But I like having this illustration here showing. I showed something similar to this in the cell signaling model of disease course. We have healthy, unhealthy, early disease, severe disease. So we have healthy. Once you're unhealthy, you have minor symptoms. Your disease starts, you have moderate symptoms. Severe disease, you have severe symptoms and ultimately maybe die. These are the symptoms ramping up. So this is showing how you have a tipping point. You're pushing a rock up a hill. This is saying it's a lot of hard work and slow momentum. But what I'm repurposing this as, throughout your life, chronic inflammation, lacking certain nutrients, poor lifestyle, the aging process, at a certain point, your cells, with the messed up cell signaling pathways, eventually you hit a tipping point where disease starts. Your healthy cells were stressed out and now they are diseased cells. The sad part is that when most people are diagnosed with their disease, it's after they already have the disease. So it's very hard to push it back up the hill. And this says lots of hard work, but it can be easy work in today's toxic world to have inflammation, to be lacking the right nutrients, to be living a poor lifestyle. So genetics can make it easier to push this up and get to the tipping point. A higher risk if you're not careful with certain weak points in your biology, but they do not guarantee issues. I've worked with people that have really messed up genes that live healthy lifestyles that don't have major issues. They just want to feel a bit better and optimize. I have people that don't have a lot of major mutations that eat like crap, they smoke, they drink, and they have a lot of health issues. So it's not just genetics. Let's talk briefly about inherited mutations, how you inherit things from your parents. Now, Mendelian inheritance. You guys may remember this Punnett square from middle school or high school biology, blast from the past here. So this means a gene inherited from either parent will go into your gametes at an equal frequency. What I mean is that I have one set of chromosomes from mom, one from dad. Every one of my sperm has one set of chromosomes. How do I know if I give mom's allele, mom's chromosomes, or dad's alleles, dad's chromosomes? It's a 50-50 shot. So this is what Gregor Mendel, who invented Mendelian inheritance, did. You have a yellow pea pod and a green pea pod. These are the alleles. So big Y is yellow. If there's one big Y, the different chromosomes, a big Y, little y versus two little y's. My point being, you draw a cross here. Half of these offspring will be yellow. Half of them will be green. Now, it doesn't mean it'll be exactly 50-50, but if you have enough offspring, it'll roughly equal out to that. Each time there's an offspring, each time you make a gamete, a sperm or an egg, there's a 50-50 chance. Now, forget about the peas though. What does this look like in humans? So here's an example here of brown eyes versus blue eyes. So dad's alleles, mom's alleles. If you have two little a's, you have blue eyes. So dad and mom both have brown eyes. But dad and mom each have one allele for blue eyes. So this means that 75% of their children will have brown eyes, and there's a 25% chance their child has blue eyes. Now, let's give a more impactful example regarding health. So let's just say these are the alleles for a gene that's linked to depression. Let's just say it's a gene that dictates how well you make serotonin. So let's just say if you have two big A's, you have no mutations. The big A means there's no mutation on that gene. Little a means there is a mutation. So two big A's, two normal alleles, happy person. This is a simplification, but just go with me on this. Big A, little a. This is half of the genes are happy. Half of them are the depressed genes. So half of the alleles are mutated, half of them are not. Double little a means both copies of this gene are mutated. So if we look at this here, with this example, mom and dad are both in the middle. They have one mutation. We look here, 1/4 of their children will have no mutations. Half of their offspring will have one mutation, so they may be a bit more prone to depression. 25% of their children will have two mutations and be at a higher risk for mood-related issues. So this is why in a family, sometimes the children may not always be as similar genetically as you may think. I worked with a lot of families, and for example, sometimes mom has 5 mutations in a gene, dad has 5 mutations in a gene. One kid has 10 mutations. He got all of them. The other kid has 0. And this is why you can sometimes have a much more sickly child versus another child, even though they were raised in the same environment. Now, I want to talk about here. So when you have the same, whether it's 2 big A's or 2 little a's, they both match. Homo means the same. Zygous, related to genetics. So what this means here, big A is homozygous. So is little a, little a. So what we could say is, this is homozygous normal. This is a homozygous mutant. If you have a big A and a little a, one of each, hetero means different. So half of their children will be heterozygous. Half of their children will be homozygous. I sometimes abbreviate these as hom or het. Now, mutations, this is really important. Mutations can synergize together. So this gene I mentioned before, the TPH2 gene, this is the gene that you make serotonin with in the brain. If you don't have this gene, you're not making serotonin in the brain. We all have it. But if someone has no mutations here and it works 100% as well as it should, then you're not at a higher risk for issues. But if you have one mutation, it may only work 80% as well. If you have two, they can stack on top of each other, or three. So the question here isn't always if I have a mutation there. Just because you have one mutation doesn't guarantee issues. But if you come to work with me, you have 3, 4, sometimes 10 mutations in a single gene. Oh, brother, this is a really big issue for you. But this isn't necessarily a bad thing, because if it's a gene we can support, we can just give you the nutrient that this gene can't make as well, and we can bypass or hack around that gene. So in summary here, the central dogma is DNA is instructions to make RNA. These are instructions to make proteins, which are the workhorses of our cells. DNA mutations always change the genetic code, but they don't always change the way that proteins function. This could be called a silent SNP or a silent mutation. DNA mutations that do change how a protein functions do not guarantee issues. Just because one protein works a little slower or faster does not guarantee you're going to get a disease linked to that, but it can increase the risk. And lastly, mutations can synergize on top of each other, meaning one cog in the machine can either be minorly messed up or really, really messed up. So I hope this gave you a good framework about the science of DNA, and looking forward to seeing you in the next module. Dr. Tyler Pansner here, and this is the Genetics 101 course. Let's talk about functional genomics philosophy. How do we take the genetics on paper into really making an impact on people's lives? So to start, what is functional genomics and how is it different from genetics? So genetics is like the study of a book that tells the story of who we are. It looks at the words or the genes written in our DNA. Scientists study genetics to understand what these instructions say and how they're passed down from parents to children. So what do the genetics mean and how are they passed on? Functional genomics is like asking, how does this story actually work in real life? It looks at what these genes are doing and how they affect the body of an actual living organism. So genetics study the instructions, and functional genomics figures out how those instructions actually affect human life. So sometimes people use the terms interchangeably, but what I really do is functional genomics. How do we take that genetic data and really get an idea of how it actually has an impact on someone's life? Functional genomics is improving people's lives through these genetic insights. It's not just what's on paper, it's what's happening in real life. Now, I mentioned this in the cell signaling model of disease course. I want to just touch on this briefly to have a better idea on how my brain looks at functional genomics. So this is a cell signaling cascade. These are 5 different classes of cellular proteins. These may ring a bell for you from the prior course. So cells receive signals from the outside world. They bind receptors. They then, it's like a relay race. They pass off the baton, and then there's relay molecules, secondary messengers. So enzymes make signaling molecules right here that bind a receptor that then get handed off to secondary messengers. And then at the end, you have a transcription factor that binds the DNA and tells your cells which genes to express more of. So that gene over there, we need more of that gene to be made into protein. That gene over there, make less protein from that gene. That's what transcription factors do. Every single one of these can be mutated in many different areas. So this is what I call building the Lego set. A lot of companies and people focus on individual mutations, a laundry list of individual mutations that are not tied together. This is what I mentioned in the introductory module about why you should care about genetics, because it's more than just the list of mutations. How do you tie it together in a cohesive plan? So by combining what we've learned thus far, we could start to build our Lego sets. So these are individual SNPs in a report just listed. But when you organize these SNPs by pathways, cell signaling pathways, then you can really get a better idea of what type of support that person needs. So, this is my unique thought. I'm not saying I'm the only one to think in this way, but I think in terms of pathways, and I really see the pathway analysis lacking in the genetic space, especially with direct to consumer testing. Now, let's give you an example of a genetic Lego set. So, this is what I would call a sleep or anxiety Lego set. These are multiple SNPs on different genes that are involved in similar pathways. So we mentioned the TPH2 gene, right? This is what creates serotonin and melatonin. This is mutated. This means the cells will make less serotonin and melatonin. Well, this clock gene, tick-tock on a clock, right? This regulates our circadian rhythm. So this is how our cells tell what time of day it is. So mutations in this gene are linked to sleep issues. Because your cells, if you have this mutation, they can't tell what time of day it is as well. So nighttime comes on by, they're ready to release melatonin, but they can't tell what time of day it is. The workers, the proteins, they don't know because they're messed up. They can't tell the time, so they release less melatonin. Now, once melatonin gets released, right? Remember, that's a signaling molecule. It has to bind a receptor. This mTNR1A, this is the melatonin 1A receptor. This is what melatonin binds to and tells our brain cells to get sleepy. This can be mutated as well. And this is also linked to sleep issues in studies because, in a different way, this doesn't affect melatonin levels, but it affects how much your cells sense the melatonin. It doesn't matter what's released in the blood. It only matters what your cells sense in the blood. Then GABA. This is a calming neurotransmitter. It slows down the brain. This ABAT gene. This gene breaks down GABA in the brain. Well, if it's mutated, it works too quickly. So it breaks down GABA too fast. There's not enough brakes to be pumped on the brain. This could be linked to insomnia. It could be linked to anxiety. It could be linked to overthinking. So you see how when you look at this Lego set, people often have all of these mutations together. So not only are they making less melatonin, they're releasing less of it at night. And the little bit they do release, they can't sense it as well. And on top of all that, their brain may struggle to calm down at night due to the lower levels of GABA. This is just one example of how you take individual mutations and compile a pathway to really give an impactful plan to really improve the quality of life of the person you're working with. Little bit more denser information. I'm going to do my best to explain it. I think it's very important for people to understand. I'm not a big math guy, guys. So we're not going to go deep into statistics here, but I'm going to try to explain this the best that I can. I want to try to make sure anyone can understand this, but it might be a little bit denser at times. Now, how do we study genetics? When I say peer-reviewed studies, what does that actually look like? So a GWAS study, a genome-wide association study. This is what's used to tell if mutations matter or not in a specific biological aspect in a given population. For example, this is a study that is correlating a certain gene. So this is called an RSID. This is a reference SNP cluster ID. This is a unique label used by databases and researchers to identify a specific SNP. So if you were to type this into Google, you would see what gene this was in and what it means. But this is a study showing the correlation between a specific SNP with Alzheimer's disease among Caucasian and Chinese populations. So what they do in a nutshell, they get a bunch of people, Caucasian and Chinese ethnicities, a bunch of people that have Alzheimer's disease, and a bunch of people that don't. And they see, is there a higher correlation of a certain mutation with Alzheimer's disease? This is an association study. So they're saying, based on meta-analyses, a lot of data together, if you have this mutation, you are statistically more likely to get Alzheimer's disease. So this mutation likely disrupts your cell signaling in a way that promotes Alzheimer's disease formation. The problem with this is that the results are in a yes or no fashion. Either it does or it doesn't. The tipping point being the mathematical level of statistical significance. What I mean by that is this is a probability curve. And you need a certain, I'm not going to get deep here, it's called a p-value, the statistical likelihood that something is significant. So if the p-value isn't low enough, that means there's a probability that this is just randomly occurring. The math we see here, it's just random that some people tend to have more or less. In a nutshell, being 95% significant or p-value, the odds of this being random is less than 5%. That right there is when people consider, yes, this matters, or no, it doesn't matter. This is the common form of evidence we have when studying human genetics, but this is limited in certain aspects that I'll discuss a little further. So again, we are living and dying by does it matter or does it not based on this number right here if we hit that value. If we hit it, it matters. If we don't hit it, it does not matter, which I think is wrong, and I'll explain in a moment. Do you think if something isn't statistically significant, it is worthless? For a lot of science, you need to pay very close attention to these values, but I'll explain more why I think it's really holding back genetics research. So, genetics matter more than people think. Why do so many genetic studies only cover a specific population? So why did this only cover Caucasian and Chinese populations? Or why did this study only study a Han Chinese population? They study this because it's a lot easier to get all of the genes or the pieces of the Lego set to align when you're more evolutionarily similar. You're more genetically similar to people of a similar race or ethnicity as yourself. So this is why one study could say in a Caucasian population, this mutation is linked to Alzheimer's disease. But in an African-American population, this is not linked to Alzheimer's disease. This is because they are only taking single mutations into account. As we mentioned earlier, a given gene can be mutated in many different areas. They're only looking at one mutation, if you have it or if you don't. But there's so much more underneath the surface of our genetics that can heavily skew this results. So if a study is done in a Chinese population, I believe it can absolutely be assumed that it is at least a contributor to all other ethnicities. We are all the same species, Homo sapiens. We are the same species of human. We all have the same genes. The mutations that we have function the same in every single one of our bodies. So even if a study, let's just say if it matters this much in a Caucasian population, in a Chinese population, I think it can be assumed that it's not that that mutation has no role in the health of any other race or ethnicity of humans. It still matters. But the way we report it is it does not matter. There is no association. Everyone thinks in a yes or no fashion. When the answer is, does it contribute at all? If it does contribute, let's try to optimize that. So that percentage that it contributes is off the table. So here's the statistical strength. Here's the significance value. So we have a Chinese population. Oh, we hit significance. It's up here. Great. But does that mean all the other ethnicities have no significance? Hope you guys are following me here. It doesn't mean this doesn't matter in another population of humans. It may not matter as much with those people in that study. Even 1,000 people that are all Han Chinese, they have genetic differences. So you study those 1,000 people, but maybe another 1,000 people of Han Chinese population, maybe it would be significant because we're genetically similar, but we're also genetically unique. This is why interpretation is so critical for genetics. Here's a study looking at two RSIDs, two polymorphisms in this NOS3 gene. This is how you make nitric oxide. This links two nitric oxide mutations to advanced chronic kidney disease and coronary heart disease in a Canarian population with type 2 diabetes. So, nitric oxide mutations linked to kidney disease and heart disease in type 2 diabetics from the Canary Islands. So, does this mean that these mutations have no role in health for other populations aside from individuals from the Canary Islands? I believe no. I believe this can be applied to other populations. This can be applied to any human being. Does this mean that these only play a role in chronic kidney disease or coronary heart disease? No, it just means in this study, the math they did proved that it hit that statistical significance. Again, this is the yes or no fashion. Once you understand what nitric oxide does molecularly, then you'll be able to grasp that no wonder why it's a contributor to these diseases. Nitric oxide helps dilate blood vessels. It helps prevent heart disease. So that makes sense. We have studies that show mutations affect heart disease in people that aren't just from the Canary Islands or type 2 diabetes. So my way of thinking is, thus, if someone has NOS3 gene mutations, boosting NOS3 gene mutations that lower nitric oxide production, boosting nitric oxide will likely help to some degree for either the disease condition across all ethnicities. Now, again, the level to which it can help different ethnicities, different disease conditions, that can absolutely vary. But when we're thinking molecularly, I do think this would be a contributor across the board. So my functional genomics philosophy, a couple of things I assume to be true. You need to have this faith in science. If a mutation is present that alters how that protein is expressed or how that protein functions. So if a mutation changes how the workhorse works, how the worker works, it will always alter the worker, regardless of your age, sex, or ethnicity. So if you have the mutation, it is always doing that thing to that gene, to that protein. Why wouldn't it? Again, this doesn't guarantee it'll cause major issues, but if a mutation is there, the mutation is doing its thing and affecting the protein. I have 100% faith in science and cellular biology, and assuming any present mutations to be true, allows me to identify the most mutated pathways. So we said before how, we had a couple Lego pieces. Well, this is how I like to look at it. You take a lot of individual mutations that on their own, people may say, you can't say that one mutation is going to cause issues. Well, I'm not saying that. I have tens or dozens or thousands or millions of mutations all pointing towards the same thing. And that's when things with genetics can really, really line up with someone's health issues. I believe supporting, how do you know which the things to support? I believe supporting the most mutated genes or the most mutated pathways allows cells to finally function as they should. If we think back to the cell signaling, right? The puppets with the strings, you can only focus on so many genes and support so many. So I like pulling the strings of the strings that are the most messed up. So simply, well, it sounds simply, but not that simple in practice, but support the genes that are the most broken. Fix what's the most genetically broken and watch the cells do their thing. That's how I approach this. Now, the level of contribution for mutations can vary. Some mutations are a lot stronger linked to issues than others, but mutations always, always, always act as they should. I wanted to talk briefly about evidence-based versus evidence-guided. Recently, we've seen a big uptick in people saying that they're evidence-based. These are people that may say you need randomized, double-blind human studies, and only human studies can be taken into account when deciding treatment options for patients. That sounds good on paper, but when you do things like I'm doing here that's ahead of where science is at overall, there isn't evidence to show if you have this mutation, you respond worse to this versus better to that. People aren't studying things in detail like that. This is a different way of thinking. I call what I do science-based or evidence-guided, meaning any piece of science or data is proof to me. So if there's a study in bacteria that is the same mutation in humans, I'll take that into account. Now, of course, the strength of these pieces of data may not be as strong, meaning a piece of data from bacteria, I'm not going to value it as much as human data, but I don't throw it out the window. Because if you have enough pieces of evidence, again, the bacteria data may not be that strong, but if you have some bacteria stuff, mouse stuff, some human things together, you can really connect the dots and really move the needle in things that other people may not have thought was possible. The significance of molecular changes varies greatly between contexts, meaning different molecular changes in how we function molecularly can vary between different disease states, but fundamental biology remains largely the same between organisms, especially mammals, meaning if there's a mutation that's linked to depression in mice, a lot of times it's linked to depression in humans. But a drug that helps mice with depression may not help humans with depression. But when we look at the genes, the genetic aspect, there usually is a strong overlap. So it's not so much if something is causing molecular changes in your body. It's how much are these changes ultimately affecting your health. It's not if your mutations are affecting your health. It's how much are they. And my approach is if I'm only using natural over-the-counter vitamins and supplements, if I have this theory about reading some data about some mouse studies and I believe that due to this mutation, someone could benefit from this supplement, it's a supplement they could already get on their own. People are already taking it. So what's the real harm in being more exploratory? I don't utilize medications. I try to get people off of a lot of medications. So what's the worst case scenario here? Someone takes a supplement. We think it's going to help. They feel a little off for a day or two. We stop taking it. They're back to normal. So this is why I love being science-based, evidence-guided. If you're too rigid about human data with everything, that's going to stifle innovation. We're not going to move the needle forward. And again, when we're utilizing over-the-counter things, we can be more exploratory. So I want to talk about, again, rigid evidence-based. They are wondering, does this factor in this study cause disease? If it doesn't, the data can't be applied to humans, meaning it's a very all or nothing type thing. Either it causes disease or it doesn't cause disease. My approach for science-based or evidence-guided, what are all of the controllable factors across many studies that contribute to disease? Address as many factors as possible, cater to that specific patient. So let's just take depression, for example. Low vitamin D, methylation issues, low iron, high histamine. These all can be linked to depression. Now, if someone has issues in vitamin D with histamine, I'm going to address multiple of those naturally. So instead of looking for the single cause, what is a contributor? And let's stack up as many contributors as we can. And here's what I like to show too, a graphic between conventional medicine and preventative medicine. So by conventional medicine, you are healthy until you get a disease. And now you're sick. And you get your diagnosis here. But something to keep in mind is clinical diagnosis is limited by technology. We can only detect cancer so early. But did you really not have cancer until you got the diagnosis? In reality, and this is what preventative medicine focuses on, this is where it's at, people. Way before you get a diagnosis, you start getting cellular disturbances. I call these the check engine lights, as we've discussed. Cellular disturbances begin. You start getting symptoms. You're feeling kind of off. You're not sick, super sick, but you're not feeling how you normally do. Then at a certain point, early disease starts. And by the time you're diagnosed, the disease is already well into the disease state, and it can be hard to reverse that. This right here, this is the check engine light area. This is where a lot of you listening right now probably are at. Our medical system, they only care when people are super sick. But that's too late, people. We need to be focusing on here, on the wellness aspect of that. Why clinical data is inherently flawed. drug, vitamin, or supplement studies, they look for correlations between the substance dose and improved outcomes. So here's a substance, right? The higher the dose, the more something changes. This can be improvements in a blood marker. So the more vitamin D you take, the more your vitamin D goes up, your grip strength, cognitive performance, self-reported outcomes, so happiness. So, for example, if you gave someone a supplement that's supposed to help with depression, the more you give them, ideally what you want to see is the higher the dose, the better their mood is, right? The more happy they feel. This is called an R, positive R. I'm not going to go deep into this. Same thing over here. The more you give of, let's say, a supplement that lowers inflammation, the lower their joint pain. The higher the dose, the lower the joint pain. Then you have a weak R association. So it's kind of more scattered, right? There's a big variance in how these people are responding to the treatment. Same thing here. Now, when you see these dots in true science, these dots should be biological replicates of the same exact thing. The biggest flaw in clinical data with genetics, each dot is a different human being that is genetically unique. We are not all the exact same in this study, so we're going to respond differently. Big Pharma is searching for drugs that help the most people any significant amount. So they just want to find a substance, a drug they can make that they could just say, yep, it significantly helps people with joint pain. They don't care about the individuality of each person. So here's an example. Here's a substance dose. Let's say this is one dose of a substance. Let's say this is happiness. It's for depression, let's say. These three people all got basically the same dosage, but look how much different their happiness is. Same thing here. Look at the huge variance in their happiness. I mean, this person got a lower dose than this person, and this person's way happier. Everyone's responding differently to these things. This is more what it looks like for clinical trials of drugs. Why do people with the same disease respond so differently to the same supplement or drug? It's because we're genetically different. So let's just say this supplement raises serotonin. Who do you think are the individuals that responded the best? Probably the ones with the most serotonin issues, because that's the signaling pathway they needed. They needed to pull the string on that puppet in that fashion. So we can see here, there's a population of people that did benefit. But the problem is, if we were to run statistics on all of these people, it may not reach that threshold. So the study says it doesn't work. But you can see there's pockets of people that benefited. So why is this a failure if it clearly helped a subpopulation of people? We should be focusing on why it did help some people and not trashing the entire thing. But that's what we do because we're trained in conventional medicine. It's trained to find one thing that helps everybody. But that's a big reason why so many clinical trials fail. It's a big reason why people are trying different mood medication to mood medication. They're just guessing because it's been shown to work for depression. How is it working? Is that the same pathway that person needs support in? For those that respond well to a given drug or supplement or treatment, the mechanism by which that works matches the disrupted mechanism of that individual's cells. So the step in the molecular biology that's working on, that's likely the right step or pathway that person needs. And if you don't respond very well, it's not that good for you. But we don't need to guess. We can be targeted with genetics. So, is clinical data only useful if it meets the significance criteria, even though it's based on non-equivalent data points? So genetically different human populations. I think it still is significant. But again, when we're judging many different people on one outcome with one mutation or one treatment, it really underestimates the power of the genetics. So here's an example. GWAS studies with one mutation are short-sighted and limited. Let's just say, all right, we're seeing that Alzheimer's study I mentioned, right? Either you don't have the mutation or you do have the mutation. This is the same gene, right? Mutant or wild type, one SNP. People can have the same SNP here, but what if this person, there's three SNPs on this gene, people. So if we're only looking at this area when we're analyzing them, this person has one mutation, this person has zero. But what if there's other mutations on this gene you're not even looking at? So now the wild type actually has more mutations than the mutant does. So that makes no sense here. It's really clouding and obscuring the data. So you can have different mutations on the same gene or different genes. So again, one step further, if we're only looking in this area, right? We're only looking here. Yes, they have a mutation. No, they don't. But this person has other mutations in the same gene. And then there's these other genes in the same pathway. So let's just say this is for vitamin D. This is for how well you make vitamin D from sunlight. Okay. But what if this is how well your cells sense vitamin D? This is how well your cells move vitamin D throughout your body. So my point being is look at the immense difference between these two individuals, but we're only looking at right here. No mutation, but it goes so much deeper than this. You could have different genes, and I made this blue for a different type of pathway. So these different strings of the puppets, they also interact together. So it shouldn't be wild type versus mutant, right? It should be further broken down. How many mutations do you have in the pathway? But we're looking so surface level. And this is what I said initially earlier on. People, these analyses are not looking deep enough for genetics. I hope throughout my career to build a platform that does this correctly because I think we're doing it the wrong way. Don't get me wrong. The current technology, we can definitely help people, but I think it leaves so much on the table. So if just one mutation in a study is linked to a disease, do you realize how much more important that actually is? It was able to overcome the immense genetic differences that we weren't taking into account. So even though they could be all so different in all these other genes that we didn't even check at, that one mutation is still significantly linked to a certain disease. So overall, what I'm saying is the way we study genetics now, it underestimates how important the genetics are. Let's just say one study says this mutation is linked to, let's say, Alzheimer's. Another study says this mutation is linked to it. Another study says this mutation is linked to it. 3 studies, 3 individual genes. But what if you have all 3 of them? If you have all 3 of them, the risk is so much higher because they're synergizing on top of each other. This is something I saw that kind of applies here. Please climb that tree. You're judging a bunch of different type of animals with different capabilities to climb the tree. Obviously, the monkey will do a lot better than the goldfish or the elephant or the penguin. So the point I'm trying to get at here, you're judging different animals on the ability to do the same task. This is like studying genetically different people, even if they're from the same ethnicity, to single genetic links to a single mutation to very complex diseases. It's not doing it the right way, if you ask me. Now, how should we cluster genes for our studies? Instead of a yes or no fashion, maybe the number of mutations in a single gene should be taken into account. So whether you're low or high number of mutations, or whether you had low, medium, or high number of mutations. So instead of you have the Alzheimer's gene mutation or you don't, maybe it's how many mutations you have in that gene. So if you go back to that example I said, we can have different buckets of people. And then maybe we see, wow, this is very strongly correlated because we broke it down further. Or maybe the number of mutations in a single pathway, whether it's low or high, low, medium, high. And this gets me into mono versus polygenic genic risk assessment. Polygenic risk score. This is the future of genetics. This is taking statistical calculations based on multiple genetic variants to give you an overall risk for a disease. Now, of course, this does not take into account environmental or other factors. It's genetic only, but it's not just one gene. It's many genes involved. So we are moving towards polygenic risk scoring, but I do think pathway analysis would be great as well. So polygenic risk scoring, basically, it's saying if based on many different genes, you're above a certain threshold, you have a higher risk. And here's a polygenic risk score. And for example, if you have a polygenic risk score of over 97.5%, based on many different genetic markers, mutations, you have a very high risk for that disease. And what we can see is that as people age, people with that set of genetic mutations do get higher rates of hypertension in this example, high blood pressure. You're more likely to get high blood pressure earlier because your genes are predisposing you to it. So instead of a yes or no, it's how at risk are you? Are you at the very low risk, very high risk, medium high, medium low, or average risk? So the more data that we come into play here, the better and clearer the picture. So these are examples from the self-decode platform that I utilize for some of my analyses. And this is showing some of the things they have, like for anxiety, they're assessing 807,582 individual mutations. These are my reports. I'm prone to anxiety. This is Epstein-Barr virus, 1,000 mutations. So some of them are lower, some of them are a lot higher. Something like anxiety, you need to take a lot more of these genetic variants into account. And now the art of this polygenic risk scoring, which genes do you use in your math models? Do you use less common? So this is how common they are. Do you lose mutations that are more common, but they may not have an effect size as much, meaning you may have common mutations that individually may not matter as much? Or do you look at the ones that are a lot rarer, that are a lot strongly linked to diseases? Because some mutations have a much, much higher risk of causing disease. The less significant each mutation is, the greater the number of mutations you'll need overall. This means more work required, but I think a lot of times people are lazy. And this is what I like to call influencer genetics. Not going to name names, but there's been a lot of companies coming out that really are piggybacking off the hype of genetics. Get your MTHFR checked and 7 other methylation genes to figure out your risk for diseases. It's a bunch of crap, people. You need so much more data than a couple of gene mutations. Even a couple dozen. Oh, I got 80 genes checked. That's nothing. That is nothing in the grand scheme of things. You're going to get very little of value checking a couple dozen mutations, or let alone 4, 5, or 6 of them. Versus clinical genetics is looking at much more comprehensive data sets. So it's not just saying fix your MTHFR, fix everything. That's clever marketing to try to get people scared about their health. But when we look more deeply, we can actually get more precise guidance. So I mentioned before, clinical trial success rates are bad. How bad? Well, here's phase 2 clinical trials are when you find the optimal dose of a medication. Phase 3 is usually 300 to 3,000 participants to see that fit the target population for that medication. So we see here, when we look at phase 3 here, under 50% for autoimmune, respiratory, endocrine, oncology, 60%, cardiovascular, a little over 50%. A lot of these clinical trials fail. Because remember, big pharma is searching for drugs that help the most people, any significant amount. They bombard or nuke cells with a single drug for a single pathway, and they hope for the best. Now, if that drug, as we mentioned, if it targets a mechanism that is involved in your flavor of your disease, more on that in a moment, but you will get healthier. Their focus is all or nothing because they're trying to fix everyone with a given disease with a given drug. I like to focus on anything that can help an individual client to the highest degree possible. So again, we're not in these studies. We're all genetically different getting this drug, so we all respond differently. So that's why the clinical trial success rates are so low, because we're all different. We're not matching it to the individual. So I wanted to talk briefly about flavors of diseases or health ailments. Each person with the same disease or label or issue can respond so differently to the same treatment because there are infinite flavors of diseases. Each person is a unique blend of their genetics. So, 5 people go to the doctor to help with their depression. Doctor will probably say, here, take a selective serotonin reuptake inhibitor, an SSRI, that raises serotonin signaling in the body, in the brain. But what if in reality, only one of them had serotonin issues? The other one had dopamine issues, or glutamate, or GABA, or histamine. You'll recall from the cell signaling model of disease course, these are different pathways that are linked to mental health issues, but all of them get a medication that raises serotonin. This is why people go on a medication. 2 people are depressed. Me and my buddy are depressed. We go to the doctor. We get put on the same SSRI. I feel great. The other person doesn't feel any different, or they feel worse, because you're guessing. What if it's a GABA issue? What if it's a histamine issue? Oh, sorry, your medication affects serotonin. Why did we pick that? Because that's what the rule book says, and we're just guessing and hoping to see what sticks. So again, depression is not just one thing. It's the culmination of multiple different cell signaling pathways. Is yours more of a histamine thing? Is it a vitamin D thing? We need to address multiple of these in tandem, not just say, oh, here you go, take an SSRI. Let's nuke your body with serotonin. It's not the way, people. And this isn't just depression. This is for any diagnosis. It's the culmination of various pathways you can control naturally through what you put in your body, on your body, or what you subject your body to. Wanted to talk about some flaws with conventional medicine. And this is what they're pretty much doing in conventional medicine a lot of the time. They're blindfolded, throwing darts at the dartboard. They're hoping this person's depressed, give them a serotonin drug. Come back in 6 weeks, we'll try something else. Not really precise here. So conventional medicine will say you're clinically depressed, take an SSRI, because SSRIs can help with depression. Well, if you look at the data, they're not that much better than placebo, but another story there. Something I would wonder, does this individual actually have lower serotonin signaling before we try to support serotonin? Because as we mentioned, there are many different things involved. So let's not assume this is the issue just because you're depressed. It could be something else. A conventional medicine practitioner may say, I like utilizing this supplement because when compared to this drug, it's just as effective as treating depression. But again, guys, the top-down approach, I like this supplement because it's better at treating depression. That's the label. That's the diagnosis. But what pathway is it acting on? You might be comparing a supplement that helps with glutamate with a drug that helps with serotonin. It's like comparing apples to oranges. You should be looking at the mechanisms and the pathways and identify which mechanism the patient needs support in. So quickly here, medical protocols, symptom or disease. They have a medical protocol where they'll guess and they put a treatment that's proven to help with your label. Let's just say they picked the right mechanism and serotonin was your issue and you feel better. Then they stop. Even though every ailment is formed from multiple factors. No one's depression is just from a serotonin issue. There's multiple things involved. So you feel better, and then the doctor says, good, let's stick with it. But in reality, you're partially healed because it's only addressing one mechanism. Or let's just say you throw the dart at the dartboard, it's the wrong mechanism. You get minimal or no improvements. Maybe you feel worse. They just guess and try another medication and give a new treatment, and they repeat the guessing over and over and over again. Gets you nowhere. My approach, you have a symptom or a disease, you analyze your pathways genetically. You identify, not guess, the top dysregulated pathways. You support those exact pathways with specific nutrients. So you're picking the right mechanisms, addressing multiple things that are messed up to get superior improvements. And this gives you improved health and resiliency. We're not just masking with a medication. We're giving the cells. We're not just blocking the check engine light. We're turning it off with the right nutrients. Now, I'm going to talk about holistic medicine flaws here as well. And let me say, holistic medicine is a big step in the right direction, but there are still some flaws here. Now, conventional, let's just say you have insomnia. They guess. They give you a sleeping medication. Band-aid fix. Why are you having insomnia? Your sleep issues are not a lack of medication. There's something else going on. Holistic or functional medicine. You have insomnia. Maybe they check. You have a mold diagnosis. They treat you with antifungal herbs or binders, and you may get rid of the mold. But why did you get the mold in the first place? A lot of people will continue to get sick with mold. But maybe the issue is that your cells are such a good host for the mold. Why are your cells so easily infected with the mold? I like to look at genetic pathway analysis. This is just a simplified example here, but let's just say there's a heavily mutated vitamin D pathway. We support that, depending on the person, how we do that. The cells can now mount a proper defense response, so they can clear out the mold. And now your cells will be much less susceptible to mold. So people I work with, it's not just getting you better now, but I hear back from you in several months or a year later, I haven't been sick in a year. Because it's not just about getting you to feel better now, it's making your cells healthier and more resilient to our toxic world so you don't keep spiraling and getting sick over and over and over again and running to the same detox protocols. And that's not a knock to these protocols. They can be great, but we need to also build our cellular resilience. Now, sometimes, and I'm not saying vitamin D is the cure to everyone's mold. It's a simplification here, but sometimes we optimize, we can't fully get rid of the mold. Then maybe we could try adding in some of these herbs if we can't clear the current infection. So we want to match the mechanism with mechanism. What do I mean by that? You want to match the exact disrupted step with the treatment whenever possible. So you have low serotonin, you have mood and stress issues. Cool. But why is it low? Can you not absorb the ingredients as well? Maybe you need more of those. Do you lack the nutrients that your workers need, your proteins need to make serotonin? Give them more of that. Maybe the worker that breaks down serotonin is mutated. It breaks it down too quickly, so it lowers. So maybe we should tell that worker to slow down. Slow them down so you don't break it down as quickly. Low vitamin D also affects serotonin levels. So if that's low, why would you ever go on an SSRI before your vitamin D is in the optimal range? It's crazy to me that people do that. Do you have inflammation? Inflammation lowers serotonin production. Do you have high histamine? That also lowers serotonin production. So you see my point here. You have mood issues. Take a drug that raises serotonin. Why is it lower? Let's just say if it's histamine. Histamine, why can't you break it down as well? These are different genes that break down histamine. When you really go granular with the genetics, is it an issue with this gene or is it an issue with this gene? Different routes for different support. So overall, functional genomics is utilizing genetics to solve real life health issues. It's not what's just on the paper, on the computer. It's how does that actually play out in real life? Build your Lego set, your genetic Lego set, to control as many genetic contributors as you can. The same label or condition or diagnosis has infinite flavors. This makes treating everyone with ADHD or depression with the same thing impossible. The thing you need to figure out for yourself is which flavor of your label are you. And genetics can help expedite that process greatly. And we don't need protocols that just fix an issue, but we also need things that help build up cellular resilience. So it's not just get rid of the mold and then I have to do a mold detox 3 times a year. What are your cells lacking? Because when I inhale mold, I don't get sick. When you do get sick. It's not just what we're exposed to, it's how well our body can fight things off. And genetics play a role in that. Dr. Tyler Pansner here, and this is the Genetics 101 course. Let's talk about ancient genetics and evolution. How did how we lived our lives, our ancestors, affect our genetics? And then how did those genetics affect the survival of our ancestors that ultimately made us us? So, the global journey of human history. So, Homo sapiens, as we evolved and became our modern species, we were residing in Africa. And around 200,000 years ago, our ancestors started dispersing across the world. Now, how did this happen initially? Well, it's thought that de novo mutations, we talk about this in the Science of DNA module, eventually made some ancient Homo sapiens more curious. It's been thought that the dopamine pathway, a lot genes involved with ADHD may have actually made certain of our ancestors more likely to say, hey, what's over there? Let's go check that out. So that curiosity drove some of our ancestors to leave. As we dispersed and settled, different traits were selected, meaning based on the environment we lived in. If you settled in an area where all you could eat was animal fat, you better make sure your genetics made you efficient at eating animal fat. So we further evolved to best suit our environments. This is why skin color also changed as we move farther from the equator. So ancestral living or ancestral dieting is a big thing nowadays, eating in according to what your ancestors did, because that's what was natural before technology, before modern man. Well, that isn't universally true. What is ancestral living for someone of African descent is different from someone from Asian descent. While we're all the same Homo sapien species, we are clearly very different genetically. So let's talk about natural or Darwinian selection. So this is Charles Darwin. You may remember him from some science lectures, middle school, high school, college perhaps. And he did a really interesting expedition where off the coast of Ecuador, we have the Galapagos Islands. And he sailed there and he realized something called adaptive radiation. So somehow, whether it's from a storm on wood or somehow animals from Ecuador ended up on the Galapagos Islands and they couldn't end up getting back. So over time, you had a common ancestor for, I think this is a finch. Over time, he saw there were multiple different species of birds that evolved. Some of them evolved to eat seeds with their big beaks. Some of them evolved to eat insects. You had this common ancestor. This is also what happened to us, a common ancestor as it dispersed here. Certain types of them. Some of them were a little more bigger beaks. They were more likely to be able to consume seeds and get nutrition and live. The whole key for this selection is certain traits in your environment make it easier to survive. You're not going to pass on your genes if you're dead, right? So another example here, different colored mice. So if we have mice that have slightly different genes, different colors, and they're black, dark mice on a dark background, dark ground, birds and predators are much more likely to attack the mice they could see easier. So these mice reproduce. A bunch of the lighter ones die. These reproduce. The next generation has less of the lighter colored mice. This is the same thing that happened with us. If we went to a certain type of environment, some of our ancestors that arrived at a certain area did not have the genes to be cut out for that, and they didn't live very long. But the ones that did had the right genes to survive better there and slowly passed that on, and we eventually morphed into different subspecies, not even subspecies, different ethnicities or races of the same species. Another example here is the polar bear. So polar bear right here, it's much harder for a seal to see polar bear versus here, right? Sticks out like an absolute sore thumb. So when the bear polar bear's ancestors initially got to these polar climates, the ones with lighter fur were less likely to be seen, so they could hunt animals better, more likely to eat, more likely to survive, more likely to reproduce. I mentioned the skin color thing here with the UV radiation. Look at the commonality here. Darker skin is closer to the equator because skin pigmentation, if you live closer to the equator, if you were born, if you have dark skin in your family, in your bloodline, and you were born in Africa and you had a mutation that made your skin lighter, you're more prone to get skin cancer. So you're more likely to get skin cancer and not live to reproduce. This also has roles for other things. This melanin that pigments our skin can be a double-edged sword. So here's a study showing the disparity in COVID-19 outcomes based on race, ethnicity, and socioeconomic status, a meta-analysis. And what this is showing is that the darker your skin color, the worse your outcomes of COVID-19. Now, I'm not going to sit here and claim that there is zero impact on the socioeconomic status and disparities in healthcare in America. What I am trying to say here is darker skin, it protects you from sunburn and cancer, but it makes it harder to make vitamin D in your skin. So this is a scientific fact. Those with darker skin pigmentation have lower vitamin D levels on average. And we know that low vitamin D has been linked to worse COVID-19 outcomes. So what I'm saying here is, I think a big factor for the disparities in COVID-19 outcomes is also the darker skin color and lower vitamin D levels on average. So some of these evolutionary things, it may help in one environment, but make it worse in another. Some of these things are double-edged swords. So the Darwinian selection, natural selection here. De novo mutations enter the gene pool. My parents gave birth to me. Let's say I'm an ancient caveman, right? My parents gave birth to me. I had a random mutation when sperm met egg. Now, let's just say I am more sensitive to adrenaline. So that means I may be more alert, more likely to see predators. So I may not die as easily because I could see predators or be more on edge. That's a good mutation in this context that enhances my survival. In modern day today, that may be a bad thing because now in our crazy, stressful world today, I'm stressed all the time. Good mutations enhance survival. Neutral ones, they'll just persist. Bad mutations will decrease survival. This is a constant cycle of trial and error. New mutations enter. Let's test ride with this new little type of Homo sapien, how it drives, right? Like a new type of car. How does it work? Oh, okay, that little mutation made them not live as long. Okay, that died off. We'll try this one. That one died off. There's all these little splits that die off if they can't make it, and the strong ones survive. So, how did genetics shape our evolution? So here's a really interesting study from 2022, the evolution of immune genes associated with the Black Death. So the black bubonic plague is caused by the Yersinia pestis bacteria. So ERAP, the ERAP gene, this is a gene involved with something called MHC2. Long story short, this is how your immune cells get activated and recognize pathogens or bacteria. This is from the study showing this is the prevalence of the ERAP mutation. So before the Black Death, this mutation was a little more prevalent. A little more of the people had this mutation than didn't. The Black Death happened. Then after the Black Death, you can see the mutation is more common. What I'm saying here is people with this mutation had stronger immune systems. This is from the study as well. Not to get too detailed here. My point being is this mutation here, the CC here, there are more immune cells in the blood of people with this mutation, whether they're not infected or whether they have the Black Plague. So my point being, when the Black Plague happened, there were a smaller number of people that had this mutation for stronger immune systems. All of the people that didn't have the mutation and weaker immune systems, not all of them, but a lot more of them died from the Black Plague. So what does that leave? That leaves a lot more people with the stronger immune systems to survive. So what we see here is right now, this mutation is higher than it was before the Black Death because it's still persisting. There's no reason for it to go away. Another example of this is amylase. So, amylase is a digestive enzyme that we release to break down sugars, break down carbohydrates or starches into simple sugars. So, a copy number variation. This is one of those chromosomal mutations we talked about in the Science of DNA module. You copy and paste a whole separate segment. So, this is showing the human amylase gene copy number variation. Now, I'll break that down for you. What this means is that if you had a higher copy number, your cells make more amylase protein. That means you're more able to break down carbohydrates into simple sugars. This means you will survive better on carbohydrates if food is scarce. So, here are individuals from different populations. So we had a common ancestor. Some of them migrated to somewhere where all they can eat are animal fat. It's a cold environment. Versus if you live somewhere more tropical where you can grow more plants, more sugar. So what we see here is that individuals living in a low starch environment, the red here, more of them have a lower copy number. The red is higher than the gray at the lower numbers. Then, as we move up higher here, you see in high starch environments, more people have that. You see more gray as you move up. What this is saying is that when you went somewhere where there was a lot of sugar available, if there wasn't a lot of food going around, and you had mainly sugar and carbs to eat, if you had more of this amylase, you got more nutrition from the carbohydrates and you were more likely to survive. Here are, again, the same thing here. These are lower starch tribes that eat more animal fat and higher starch tribes that eat more plants and carbohydrates. And we see here more of them, this is shifted to the right, the high starch. So it's showing kind of the same thing here. Don't want to get too nitty gritty here, but my point being is our genetics, where we go, what we're eating dictates what types of individuals will survive better. And over time, that trial and error process, you'll produce populations that have pretty significant differences in their genetics. So a question I get very often is, well, if these mutations are so common, then how do we pass this on? Another common question I get is, how bad are my genes versus other people? Well, certain mutations, we kind of touched on this, certain mutations may have helped survival in some areas of the world while hindered it in others. So again, having dark skin, high melanin, will help you if you're exposed to a lot of UV radiation. But if you're not exposed to a lot of UV radiation, it can also lead to a vitamin D deficiency. The mutations I like to focus on are quite common, which means each individual mutation may not be a massive factor, but this is why you need to build your Lego set and string multiple of them together. This is important too. Genetics that allowed survival and reproduction only really mattered at younger ages. I mean this because our ancestors, all they had to do was live till their teenage years to reproduce. They did not have to live a long, healthy life now, 70, 80 years old. So my point is, genetics that may be more negative for us today. How did I get all these bad genes from my ancestors? Well, all your ancestors had to do was make it till 13 or 14 or 15 and have intercourse, reproduce. And then they pass on their genes. Our toxic modern world is ultimately what is driving up the rates of chronic disease. Our genes have not changed much over the past 200,000 years. But our genes, we can't do that much about our environment nowadays. Sadly, the air we breathe, the food we eat, the water, it's very, very hard. So even though you can't pick your genes, you can figure out where your biggest genetic weak points are and address that appropriately. And again, when I said these are quite common, some of these mutations I focus on, over 50% of the population has them. But again, you need to line these up and map them out appropriately. So Did cavemen get diseases? This is an important point too. People tend to act like our ancestors did not get any modern diseases whatsoever. So here is when modern humans formed 100,000, 200,000 years ago or so. We can look back. Genetic diseases, we have proof of this from 4 billion years ago. Before we were even multicellular organisms, genetic diseases existed. The second we became multicellular organisms, history of cancer. We have history of autoimmune diseases. We have history of schizophrenia. We have history of schizophrenia soon after we evolved away from chimpanzees. So my point is, while yes, you look more recent, obesity, type 2 diabetes, autoimmunity, allergies, these are more recent things in our evolution. But people act like there weren't any genetic issues for our ancestors. Remember, imagine like a tree splitting off. You could have some mutations enter a population. They keep reproducing. Eventually, the wrong mutations happen. They get these diseases. They pass away. So the overall take-home message here is that messed up genetics have been affecting not just Homo sapien health, but all of our ancestors for billions and billions of years. Genetics have played a role in our evolution, our health, and our death as we evolve. So overall take-homes here. Darwin's Galapagos animals is fairly similar to our ancestors spreading across the globe. As we dispersed, our genetics shaped how we survived or didn't survive. So this dictates which mutations are passed on. Our modern environment is the true cause of rising illness, and it's targeting those with higher genetic risk. If you think about it, if we're all exposed to mold, who are the people that are going to get the sickest from mold? Probably the people with the worst genetic defense against mold. But our ancient ancestors also got diseases that we suffered from today. So it's not like all genetic stuff is a brand new thing now, but it's all about balancing how messed up are the genes, how toxic is the environment.